Welcome to our exploration of genomic variation, a crucial aspect of genome biology. Today, we'll uncover the significance of genomic variation, identify its types, and explore detection techniques and real-world examples. Genetic variation analysis spans from bacterial traits to human disorders, comparing genetic makeup across species. Genomic variation arises from mutations, recombination, gene flow, and natural selection. Recombination involves genetic material exchange during cell division, crucial for diversity. Homologous recombination is a key genetic mechanism, crucial during meiosis for genetic diversity. Gene flow transfers genetic material between populations, enhancing variation. Genetic drift causes random allele frequency changes, impacting small populations significantly. Genomic variations include SNPs, indels, CNVs, and structural variants. Polymorphism involves stable allele existence, while mutations alter base sequences. SNPs are simple genetic variations that can affect protein function. Techniques like PCR, microarrays, and sequencing help analyze SNPs. PCR amplifies target DNA for sequencing, crucial for SNP analysis. Microarrays use probes to analyze thousands of SNPs efficiently. SNP arrays compare SNP presence with specific traits in populations. Affymetrix and Illumina use different probe lengths for SNP analysis. SNP arrays help visualize genetic variations across individuals. Larger genomic variations include insertions, deletions, and rearrangements. Insertions add nucleotides to DNA, affecting genetic sequences. Deletions remove nucleotides, impacting genetic sequences significantly. Microdeletions and macrodeletions are crucial concepts in genomic variations. Microdeletions involve small segments of DNA, while macrodeletions affect larger chromosome sections. Both can significantly impact genetic information. Indels, or insertions and deletions of DNA segments, play a significant role in genetic variation. They are more prevalent than single nucleotide substitutions and often occur in protein coding regions, suggesting strong natural selection in human evolution. Copy number variants, or CNVs, are segments of the genome that differ in the number of copies due to duplications or deletions. They contribute to genetic diversity and can sometimes lead to diseases like Huntington's. Huntington's disease involves the repetition of 3 base pairs in the HTT gene. In healthy individuals, these repeats range from 10 to 35 times, but in patients, they can vary between 36 and 120 times, illustrating the impact of CNVs. CNVs can also describe repeats of whole genes, such as the alpha amylase 1 gene, which breaks down starch into sugars. This gene can appear multiple times in a genome, with some individuals having up to 14 copies, especially in high-starch diets. Genomic variation leads to evolution and diversity. Different types, such as SNPs, indels, and CNVs, have varying impacts. Tools like sequencing and arrays are essential for studying these variations. Consider which type of genomic variation you found most interesting and why. Reflect on how this knowledge could be applied in medicine or research. Here are some references for further reading on genomic variations, including studies on SNP arrays and indels in coding regions. Thank you for your attention. Are there any questions? Welcome to our session on analyzing genomic variation, focusing on structural rearrangements and sequencing techniques. Let's dive into the fascinating world of genome biology. Today, we'll understand structural genomic rearrangements, explore a case study on the Philadelphia chromosome, and delve into genome sequencing techniques like Sanger sequencing. Structural variants, or SVs, refer to DNA segments larger than 1 kb that include inversions, translocations, insertions, and deletions. These are commonly known as copy number variants, or CNVs. An inversion occurs when a chromosome segment breaks off and reattaches in reverse order. This can lead to significant genetic changes, although DNA may not be lost in the process. Translocation involves a chromosome breaking and the fragmented pieces reattaching to different chromosomes. This can have profound effects on genetic information. Studying structural variants is crucial for diagnosing certain genetic diseases, as they can reveal significant insights into genetic disorders. The Philadelphia chromosome results from an exchange between chromosomes 9 and 22, leading to chronic myeloid leukemia. This fusion creates a protein that affects cell division. DNA sequencing is a laboratory technique for determining the exact sequence of nucleotides in a DNA molecule. It's essential for understanding gene functions and genome parts. DNA sequencing techniques have evolved, with costs dropping significantly over the years. Ongoing research continues to enhance these methods, making genomics more accessible. Frederick Sanger, a Nobel laureate, developed a DNA sequencing method that revolutionized genomics. His work laid the foundation for modern sequencing techniques. In 1977, Sanger introduced a reliable DNA sequencing method using chain-terminating molecules. This marked the first generation of sequencing technology, paving the way for future advancements. Sanger sequencing is a method used to determine the sequence of DNA. It requires a single strand of DNA, a primer, DNA polymerase, and special building blocks called DNTPs. Additionally, special stop building blocks known as ddNTPs are used to terminate the DNA strand, allowing for sequencing. The Sanger sequencing mechanism involves dividing a DNA sample into 4 tubes, each containing all 4 normal dNTPs and 1 special ddNTP. The results are read by running the reactions through a gel, which separates the DNA fragments by size, allowing the sequence to be determined. Quickly understand Sanger's sequencing with this visual overview. It simplifies the process into key components and steps, making it easier to grasp the essentials. The history of sequencing technology spans from the Sanger method in 1977 to the third-stage Human Microbiome Project in 2019, showcasing significant advancements in DNA sequencing over the decades. In this group activity, students will explore different generations of DNA sequencing technology. Each group will focus on one generation, with roles assigned to ensure comprehensive understanding and presentation. Guidance questions are provided to help explore the basic understanding, methodology, and strengths and limitations of each sequencing generation. These questions will guide the research and presentations. Whole genome sequencing, also known as shotgun sequencing, involves cutting DNA into short segments, sequencing them, and aligning them to form a complete genome sequence. The alignment process in whole genome sequencing uses software to compare sequences and identify overlaps, allowing scientists to reconstruct the genome. This method was notably used by Tiger in 1995 to sequence the genome of Haemophilus influenzae. Sonication of DNA involves using ultrasound to shear DNA into smaller fragments, which is a crucial step in preparing DNA for sequencing. For further reading, explore these references on structural variation, the Philadelphia chromosome, next-generation sequencing, and more.